Welcome to this explainer. I'm so glad you're here because today we are diving into one of the most stubborn medical mysteries out there, hepatitis B. As a research scientist, I can tell you this virus is an absolute master of disguise. But we're not just here to talk about the problem. We are looking at a massive, game-changing breakthrough. We're going to explore some incredible genetic tools, synthetic, precision-engineered molecules that are quite literally rewriting the rules of how we fight viral infections. If you've ever wondered how modern science can intercept a virus's genetic blueprint right before it causes harm, well, you are in the exact right place. Let's get into it.
Okay, let's dive into this. 240 million. That's the estimated number of people globally living with chronic hepatitis B right now. It's a staggering number, isn't it? And look, this virus is definitely not just a benign passenger in the body. When someone is infected, especially early in childhood, the virus zeroes in on the liver, leading to chronic disease. This puts millions of people at a severely elevated risk for devastating liver problems. We're talking severe scarring like cirrhosis, total liver failure, and hepatocellular carcinoma, which is primary liver cancer. All told, this is a disease claiming over 1 million lives every single year. So the stakes for finding a true cure, they literally could not be higher.
And this brilliantly illustrates exactly what we're up against. To understand why this virus is so incredibly dangerous, and honestly so difficult to eliminate, we really have to look inside the liver cell itself, the hepatitis. Once the virus gets inside, it uncoats and ships its genetic material straight into the nucleus. And you know, the nucleus is the absolute control center of the cell. Once it's in there, it undergoes this repair process to form something called cccDNA, or a covalently closed circular DNA. Now, I want you to think of cccDNA as the virus's hidden master copy. It firmly drops anchor in the nucleus and acts as this continuous churning template, just pumping out more viral RNA and toxic proteins. It's essentially a rogue factory that simply refuses to shut down.
So, to understand our current biological roadblock, we have to look at what we're actually using right now to fight that rogue factory. Today, our standard go-to treatments rely mostly on nucleoside or nucleotide analogs, we usually just call them NAs, along with pegylated interferon. Now, to be fair, these drugs are fantastic at doing one very specific thing: suppressing viral replication. They stop the virus from actively multiplying, which definitely improves liver inflammation and slows down the disease. But here is the massive catch. They leave that hidden master copy, the cccDNA, completely untouched. Because the main factory is still intact, patients achieve what we call a functional cure, where the virus is virtually undetectable without meds, only about 1% of the time per year. For the vast majority of people, this means being tethered to lifelong therapy. No one wants that.
Now, you might be wondering, wait a second, didn't we cure hepatitis C? Why is B so incredibly different? Well, it's really a tale of two viruses. Hepatitis C is an RNA virus. It replicates out in the cytoplasm of the cell, just completely out in the open. Because of that, our modern direct-acting antivirals can just swoop in and essentially wipe it out. Boom, patient cured. But hepatitis B, that's a DNA virus. As we just saw, it sneaks into the fortress of the nucleus and drops anchor with that cccDNA. It is a total master of disguise, evading traditional treatments by literally burying its blueprint inside our own cellular machinery. So while we can cure Hep C today, up until now, we've really only been able to manage Hep B.
But what if, and this is where it gets exciting, what if we could intercept the messages coming out of that factory? Enter antisense oligonucleotides, or ASOs. I want you to think of ASOs not as your standard everyday pill, but as precision genetic scalpels. Look, since we can't safely destroy the cccDNA hiding in the nucleus without risking the host's own DNA, we target the temporary copies it sends out instead. ASOs are these incredibly cool, short, highly engineered synthetic chains of nucleic acids. They are specifically designed to seek out and bind with absolute precision to a target RNA. They literally hunt down the blueprints the virus uses to build its toxic proteins.
Let's move to and see how this builds, because the real beauty of ASOs lies in their exact three-step mechanism of interception. Step 1. The ASO enters the cell and uses classic Watson-Crick base pairing to bind specifically to the target hepatitis B RNA transcripts. It is a perfect genetic match. Step 2. Once it binds, it creates this double-stranded structure. Our cells recognize that structure as unnatural, which actually triggers our own cellular enzymes to come over and actively degrade that viral RNA blueprint. And step 3. Without the blueprint, the whole manufacturing process grinds to a halt. The virus can no longer produce its toxic proteins, including the hepatitis B surface antigen, or HBsAg, which is what normally helps the virus hide from our immune system in the first place.
Applying this brilliant technology specifically to hepatitis B brings us to a revolutionary drug currently in development called bepirovirsen. So the crucial point here is that bepirovirsen isn't just any ASO, it delivers this totally unique 1-2 punch that makes it incredibly promising. First, you have its direct action. Just like we've discussed, it targets all those HBV RNA transcripts, degrades them, and drastically lowers the levels of the virus and its surface antigens. And by lowering that surface antigen burden, the virus can't maintain its immune tolerance anymore. But then, and this is huge, comes the indirect action. Bepirovirsen actually stimulates the host's innate immune system. It wakes up the body's natural defenses, causing immune cell activation. It literally starves the virus while waking up the guard dogs at the exact same time.
Now, theory is great, right? But as scientists, we want to see real-world clinical impact. And the data here is just remarkable. In the Phase 3 B-Well-1 and B-Well-2 clinical trials, researchers tested 24 weeks of bepirovirsen therapy on patients. The primary outcome they were hunting for was a functional cure at week 72. Now remember our historical functional cure rate with standard therapy? It's right around 1%. Well, in these trials, the percentage of specific patients achieving a functional cure at week 72 was approximately 20% with bepirovirsen, compared to absolute 0% with the placebo. Let that sink in for a second. We are looking at a massive leap from a 1% historical baseline to a 20% functional cure rate for these specific patients. That is a total paradigm shift.
Of course, as researchers, we must always maintain our scientific objectivity, which means we have to talk about safety. The overall safety profile for bepirovirsen in these massive global trials was deemed acceptable and consistent. However, the most common severe adverse event they observed was an elevation in ALT, which is a liver enzyme. Now, usually, in the context of liver disease, high ALT means liver damage, which sounds bad. But in this specific scenario, researchers note that these ALT flares might actually be a fascinating sign of success. They may indicate immune-mediated clearance of the virus. Basically, it's the newly awakened immune system aggressively attacking those infected liver cells. It's doing exactly the job it was supposed to do all along.
Now, what's really interesting here is how this fits into the broader picture of treatment. While bepirovirsen is a massive breakthrough on its own, the future of medicine for hepatitis B is almost certainly going to be a finite combination therapy. The ultimate goal requires us to target the virus from multiple angles simultaneously. We need drugs that inhibit viral replication, like our current NAS. We need drugs that lower the viral antigen burden, like ASOs such as bepirovirsen. And we need to boost the host immune response. Notice how perfectly bepirovirsen fits into the intersection of these targets? It impressively covers 2 of those 3 requirements all by itself. By mixing and matching these targeted therapies, we can finally close the net on the virus from every conceivable angle.
So we have to ask the ultimate question based on all the evidence we've seen today. Are we finally entering the era of a functional cure for hepatitis B? The incredible science behind antisense oligonucleotides shows us that we're no longer just trying to quietly suppress this virus. We are actively disarming it at the genetic level and rallying the body's own defenses to finish the job. Bepirovirsen represents a massive leap forward in precision medicine and hepatology, offering real, tangible hope to millions of people. The master of disguise has finally been unmasked. The only question now is, what other viral giants will this ASO technology help us conquer next?